this is Jessica DeMassa in the GuideWell Insights Lounge here at Singularity University's Exponential Medicine. I'm joined by two very special guests. We have our co-host, John Mattison, who's the Chief Medical Information Officer for Kaiser Permanente, and we have our interview subject, Andrew Hessel, who's a distinguished research scientist with Autodesk. So I'm going to turn this right over to you, Dr. Mattison, so feel free, fire away.
It's an honor because I've gotten to know Andrew for a while, and I just so impressed with what you've been doing. But one of the things that keeps coming up in this conference is how the greatest disruptions that occur in any particular industry often come from outside of the industry. So when we think of Autodesk, we think of CAD/CAM software, we think of the space shuttle, we think of all the early work, but you're actually getting very deeply involved in healthcare with your 3D printing and all of the work that you're doing there. So tell us a little bit about why your company decided that there was a trajectory that you wanted to take into healthcare. How did that discussion occur?
Well, it started with a conversation that actually came out of our early support of Singularity University, and this was it was injecting a lot of new ideas into the company. It also brought some new people into the mix. One of those was me, and I walked into we have a gallery space in San Francisco that showcases not the software, but what people make with our software. And after reviewing the gallery, I went up to the CEO and I said, everything in here is amazing. Like the work that the company does with its partners is really breathtaking, but it's all large and it's all non-living. Would you be interested in learning how to design living things? And he was really receptive to that idea, started an investigation and some diligence in that. And we realized there were no tools in the company to work at these new domains, the nano or the micro, or with these really amazing living systems, which you can think of as self-assembling machines. So after a couple of years of diligence, they founded the BioNano Group in 2012, and we started looking at opportunities to make a difference and to apply our expertise in design and engineering and media into these new and unfamiliar areas.
And a lot of people would think, wait a minute, so you're talking about 3D printing, that's physical, but we think of humans and the human experience as being very complex biologic systems that are all interacting with each other, and we think of 3D printing as a physical solution set. So how do you cross that chasm from a effectively purely physical 3D printing into the biologic realm? How do you conceive of that? Is there a single pathway? Are there multiple pathways? What does that look like?
There's multiple pathways, but at the core, what we have to remember and what we see so much of at this conference is life science, and the world around us is being digitized. So that's our play space. Autodesk makes digital tools and digital manipulation tools. So what we had to focus on was the areas kind of bottom up. You know, we were adept at designing and building larger things that have conventional physics, things like buildings and cars. So we had to go into this unfamiliar world of molecules and then polymers, strings of molecules, things like proteins, or DNA chains. Up from there to these molecular machines, what we think of as nanoparticles, viruses or engineered particles. And after that, you're starting to work with living cells, these complex systems, and you're either reprogramming their metabolism or you're completely building their genomes from scratch. Once you have those materials, once you have the living cell, which you can think of as a 3D printer, except it doesn't make one, isn't just printing with plastic or metal, it's printing molecular structures from scratch, elemental materials, and doing thousands of different materials at once, which is the complex problem. But once you've got the cells, now you can use that as a substrate for traditional 3D printing, where you're using some sort of print head or synthesis system to do additive manufacturing. You can actually have cells be part of that. So basically, bioprinting is a crossover point. Whether you're doing biomaterials or living materials in a printer, that's our crossover point where we can start to talk to the rest of the company and their familiarity with 3D printing and larger manufacturing systems. So we look at the future of manufacturing, and what I love to say is the future of manufacturing is really self-assembly and growing the things that we need for humanity. Part of that is medicines, but it can be materials as well.
That's a really fascinating way of looking at it. Is sort of the human cell is sort of the ultimate nanotechnology for synthesizing just a wide array of things. So it kind of raises the question, if the human cell is so evolved and so pluripotent in terms of its synthetic capacity, what do you see as boundary conditions? What do you see as things that represent big challenges in terms of how you begin to replicate some of those features and functions? How do you tackle those issues incrementally in a way or exponentially in a way that you get to where you want to go?
Well, that's a great question, and I just want to clarify one thing. It's not just human cells. All living systems, whether it's plant or animal, we're all made of cells, and human cells are not that different from cells of a dog or even a mouse. So the basic architecture is conserved. But what we started to realize is we have to start getting more access to this data and understand it. So this is where systems biology and omics, Lee Hood spoke the other day, is really important, because now we're digitizing the entire mechanism of the cell and getting more of that information available for computation analysis and eventually design. And then on the design front, we have to be able to translate our intentions into code. Not digital code, zeros and ones, but genetic code, A's, T's, G's, and C's. And this has been a field that is emerging, synthetic biology, where we are really coding living things. But so we knew we had to start focusing on design tools there, but kind of the next generation design tools, taking it bigger and better. And part of our work there, we ended up helping to sponsor and organize scientists and catalyze what we think of as the next big genome project, being able to think about not designing a single protein or a metabolic pathway, but building the tools and technologies for designing and writing entire large genomes for cells.
Well, thank you so much, Andrew, for that wonderful answer. It was a pleasure to have you here and to speak with you. And John, thank you so much for stepping in and asking such brilliant questions, because I know I could not have asked those. So we want to thank you both for your time again and for joining us here in the GuideWell Insights Lounge. I'm Jessica DeMassa here at Singularity University's Exponential Medicine.